and I'm also the Chief Scientific Officer of Northern Therapeutics, which is a small Canadian company that's targeted in developing cell-based therapies for acute diseases such as pulmonary and chronic diseases such as pulmonary hypertension and septic shock. So we have a number of interesting clinical trials sort of taking place in that field I'll talk a little bit about today. I also sit on the CTSG part of CellCan, so I take any issues to Health Canada. We meet twice a year, as Leah talked about as well. And I'm also a regulatory consultant for the Ontario Institute for Regenerative Medicine. So what I'm going to talk about today are some things that not directly involved in how we sort of operate our clean rooms, but things that may, coming from outside, that have a big impact on the quality and the safety of the products. So that's the title of the talk today, is Extraneous Considerations for Cell and Gene Therapy Manufacturing. So just to start off, if we have this slide, which I pulled from the web, is interesting, where we see in terms of the stem cell development of stem cell therapies, the typical growth curve where there's this big peak where a lot of interest takes place, and then there's a bit of a drop-off where some of the first products were approved, and Carticel and the ESC is isolated. And then there was a bit of a drop-off in 1999. Interesting, 1999 also corresponds with the Jesse Gelsinger case, which was the first death that involved gene therapy. So that had a big impact both on the gene therapy field, and as well as we can see there's a drop-off in interest and approvals for cell therapy-based products as well from that initial excitement. So we went into a bit of a lull in the early part of the 21st century, and then things have started to slowly grow back up. And I think now we're at an inflection point where we're getting really an extensive amount of excitement involving the cell therapy, and not only cell therapy, but gene therapy is now taking off like I've never seen before. And many of the cell-based gene therapies are very exciting and very interesting. And there's a combination of these products that seems to be exciting. And of course, when we look at that, one of the major products that's leading that field is, of course, the CAR-T, which has been driving the field now for about 8 or 9 years. And it was surprising after the Jesse Gelsinger case how quickly these retroviral transfected cells came onto the market. And the first CAR-T therapies look very exciting, very, very, very promising. And I think now we're becoming to that point where where are they going to fit in as a marketable product? And are they going to be first to market means first to disappoint? There's some conversation and some interesting articles around that. And part of the issue is the cost of the products, that what they're being charged at, and the revenues that these companies are actually generating certainly are far less than they would have expected. And there's a number of reasons for that. And part of that involves some of the manufacturing and the constraints on pricing and reimbursement that are taking place throughout the U.S. makes it difficult for them to get substantial market share. But there also has potential issues with these products in terms of relapse and other issues. So as we're moving along, what we're seeing is a rapid development in this space and a massive investment to move these products forward, which means for us as academics developing clinical cell therapy products, there's a lot of excitement and there's a lot of opportunity, but I think there's also a lot of concern that we have to do this in a very high quality manner with evidence-driven experimental medicine being the underpinning concept that drives the field forward. And there have been, I think, as we can see here, some Novartis has run into some production problems. And those production problems, again, something I'll touch on, I'm not going to spend a lot of time in this talk, is the source product. And the variability of the source products makes a consistent production of the autologous product somewhat difficult, but not impossible. And just means you have to do it very carefully. You have to understand and spend a significant amount of time understanding the initial starting product and do your best to reduce that variability in the starting product so you can make a consistent product at the end that's like to be consistently therapeutic. So of interest, if you look at the FDA summary basis for regulatory action, the regulatory action in August 2017 was the approval of Chimera, which was the first T-cell CAR product to be approved in the States. by from Novartis. They summarize the manufacturing controls or control strategy. And the first on that list is raw material and reagent qualification programs. And that's so to maintain the manufacturing process consistently is mainly controlled really by what you're starting with. And then then after that, you look at your in-process monitoring, in-process control testing, including the decision points for alternative manufacturing pathways, your lot release tests, traceability by use a chain of identity system, so traceability all the way back to the donor, and validation of your manufacturing processes. But all of these really rely on that starting material that you're putting into the system. So the raw material qualification program that Novartis got approved on consists of a risk assessment of the source materials, the vendor qualification and audits, and an incoming materials management system that consists of visual examination of the package, identity testing, and confirmation of certificate of analysis upon receipt of the raw material and the reagent. So really this is what I'm going to focus most of the talk on is how do we do that? How do we make sure that the raw materials that are coming into our CGMP facilities are actually of appropriate quality and validation and are consistent enough when we store them there that they can be used to manufacture a high quality cell therapy or cell-based cell therapy gene product. Not surprisingly, this is a slide I like to show a lot, and that is that when we move through the approval phase for a cell-based product, it's very similar to a pharmaceutical as well. Of course, we start with basic research and the disease discovery and the drug discovery, and then the preclinical development. And this way, a cell therapy would fit into this. And moving then towards GLP, usually we do certainly toxicology studies in animals at a GLP level to ensure that the cell therapy product is working well. And there's a number of issues with that are beyond the scope of this talk today. We have to have some GCP involved when we start our clinical trials, which is good clinical practice. And those clinical trials, although classically for drugs it's phase 1, 2, and 3, what we've really been seeing with Health Canada, the definition for cell therapy products are more like early and late phase trials. So the early phase would be first in man, you're testing really the safety of the product, and then almost all of the efficacy trials become lumping in this later phase process. There usually isn't a phase 1 clinical trial where you're just testing in healthy volunteers. That's really, I don't know one that has been done in a cell therapy trial. So we're almost always having some disease patients, and we're looking at efficacy, although it's not the primary endpoint. Even our early phase trials be focused on safety. And then as we move through these clinical trials from early to late, it's the rigor in that GMP that needs to go up. When you have a licensed product, you have to be really full GMP, and that is the definition of the GMP process. It really applies to the licensed product to ensure that that licensed product, no matter how you're producing it or where you're producing it, is actually of a high quality of what you had in your submission, in your regulatory submission. But when we start in the early phase 1 trials, we may not be at that very high level of GMP quality assurance. There's a number of reasons for that. The first and foremost is that often when we start these phase 1 clinical trials, certainly from an academic basis, the trials are being done to learn as much as we can about the product, particularly if we're thinking of an autologous product. We may not be taking the cells from a number of diseased patients prior to going into a clinical trial because we thought it was unethical to do that in a patient and not give them a potential of some therapy. So in those cases, we're learning about what the initial starting product may be like, what the variability is, how consistently we can make the product. So there has to be a little bit more flexibility in the process because the early phase trials in cell therapy products are often very much a learning phase. But as we move through those phases, we should have really started to lock in our process, have a validated process, and really have a tight control of all the ancillary products because they should be well defined in what we're placing on that product so that you ensure both consistency in terms of potency, and quality of the product that's coming out of the facility. So in this variability as you move from early phase to late phase really is something that's very difficult from both a regulatory point of view, because the regulators can't say exactly what you should be doing and when you should be changing, and certainly from someone trying to design a clinical trial, design a processing for this clinical trial, because you're beginning to look at what's going on there. So what is GMP and why do we need it? Well, good manufacturing practice guidelines set out the quality standards for manufacturing of medical products. They aim to make sure products such as drugs and vaccines have consistent strength, purity, and quality. And that's what if they're not consistent, the variability in clinical trials as you start out and you're adding to that variability, is unlikely you're going to see a positive result. So GMP really involves the infrastructure, the buildings, the clean rooms, how they're built. Are they built adequately to provide this isolation? The staffing, the training of that staff so they can operate in those clean rooms, the equipment, the monitoring, and the processes that you've got, so all your SOPs, the sanitation protocols that you have in place to make sure the room and all of your processes are done in a very clean fashion. But also what you're putting in, which is what goes into that room, which is largely your ancillary reagents or ancillary materials. And then we have quality control and quality assurance on top of it. And it all involves validating all of these things and documenting everything. Now, cell therapy products are unique in that most represent a subset of products that cannot be filter sterilized and therefore undergo aseptic manipulation through the manufacturing process. In fact, in most cases, cell therapy products are defined by the process, not the end product, because the end product is almost next to impossible to completely define on a chemical basis, as you would a drug. So the cell therapy product, which likely could be highly variable at the end, maybe a mixture of lymphocytes or other T cells as well, is not an exceptionally high defined product, but is defined by the process that product underwent to manufacture it. So that's where we have to be very careful in terms of the process. In some instances, the FDA has said where possible, closed systems should be used during manufacturing. I think I'll touch on that a little bit. We're moving away from these large clean rooms and more to closed systems, which is great, but that still leaves us with the input that's going into those closed systems. And that's what we're going to talk about a bit more today. So, but still, for most of the rooms that are out there, most of the products that are out there, the classic processing is in an ISO 7 room, which I believe would be equivalent to a Class B clean room, with all open processes performed in an ISO 5. In other words, a very clean environment, that's a laminar flow hood, or an isolator where you're defining all of your closed processes. Other closed processes, we know, and in Kingston, Octane has developed this system, which Lonza has now adopted as a closed process for manufacturing adherent cells, such as MSCs could be manufactured in these pods. It's a completely closed process. We now have the Terumo Quantum, which is a closed bioreactor system. And of course, the Molteni Prodigy, which has been specifically designed for the development of T-cell CARs or other gene-modified lymphocytes, has now been widely adopted in a number of academic centers around the world. Although the first clinical trials using the Molteni are underway, none of those have been reported as yet. But a number of these systems are out there, and the advantage of it, having a closed system start to finish, is highly desirable. But does not mean that you cannot eliminate all your staff. There still is a great deal of staff involved in the operation of these systems. And as you can see, there's a number of reagents and also hard materials that are disposables that are involved in the development of these products, even on a Prodigy. And that's where the qualification process really needs to be very carefully done. And not only qualification of the incoming materials, but how those materials are then stored and validated storage procedures and stability of those products while you've got them in your facility prior to using them. So this just shows that some large-scale autologous cell production can be scaled out very nicely with these systems. So multiple systems like this can be placed around the country and around the world now. I think Molteni is in almost in most continents. There are Molteni systems and people are using them. So if we look at the 21st century GMP to support autologous or bespoke cell therapy products, we call bespoke that would be specifically designed to the patient. We have limited infrastructure for wide geographical distribution, but it can be cost effective, enhanced consistent product quality through precise control. Isolation for aseptic processing should be risk-based, science-based, evidence-based, and we should continuously be looking to improve the processes and qualities that we're implementing to put these things forward. So improperly designed clean rooms and or closed processing system used with appropriate operating protocols, the highest risk to product safety and quality come from the externally manufactured reagents, materials, and devices, as well as your starting cell product that's going in. So if we look at moving from the older systems that we have out there now where there's extensive amount of gowning, both of which are very safe and effective, the newer systems may require much less gowning and easier operating environment to an extent, but in working in isolators where the equipment and even your closed systems could be done in an isolator environment, which is completely ISO 5 and can completely independently sterilize from the rest of the room, so making changeover a little bit easier as well. So we've adopted these biospherics isolators at OHRI. We feel there's a big advantage to processing at least early phase clinical products in these devices because it allows us to gather a whole pile of information, environmental information on the growth of the product that we don't get by using external incubators. So again, it's the highest risk of the quality we think is going to come from those external materials, and that's what we want to talk about now. So first of all, let's talk about the cell source. Cell sources can be autologous, in which case the donor eligibility determination is not required, although you might have some exclusion criteria within your clinical trial, particularly in an early phase clinical trial. We would often have the exclusion of very highly infectious diseases, and when we're treating our chronic diseases, it just causes complications, not only and the biggest risk, of course, is the infectivity that may then apply to both either staff working in the clean room or the possibility of transfer to other autologous products that are working in that room. So if you do have highly infectious patients coming into your trial, you certainly there have to make a concerted effort to make sure that there's no chance at all that there could be cross-infectivity between the autologous donor samples. And a lot of that would likely be down to spatial isolation and temporal isolation, one patient cells in any one room at a time in a complete changeover protocol that would be proven to kill the infectious agent involved. We do have the issue of labeling in autologous products, and the biggest, a big safety issue in autologous products, of course, is making sure that the right cell goes back to the right patient. So that's a lot of labeling requirements, and tracking is essential for autologous products. Processing is shown not to support propagation of infectious agents is highly desirable in many of these cases. Some of that can be done with antibiotics involved or antifungals involved in the processing, but it's not necessarily the only thing that needs to be involved. If it's a long-term process, you could also make sure the products are clean right from the start to the finish. But if it's a short-term autologous therapy, you often won't get your infectious disease results back from either bacteria or virus prior to the product going to the patient. So that's often not available to you. In allogenic, that is because often you're making larger cell banks, and that's where you need both the donor screening and testing because you want to make sure the product is coming in as clean as you can. In Canada, we do it to the cell tissue and organ guidelines, which has a specific testing outline for the adventitious and bacterial agents that you'll be testing every of your donors for. And also you have to appreciate that those donors should be tested within a specific time of harvest or collection so that there's no chance that they could be reinfected or infected between your testing and your harvest from them. So, and there's an exception to that in I think the US FDA, and I think also in Canada for some stem progenitor cells collected from the bone marrow, but it's only up to 30 days and not the 7 days. In both auto and allo samples, of course, the original cell product should be retained for future testing if necessary. If you do come back with a positive results in some of your testing downstream where the patient gets an infection, it's often nice to show that it was not in, or was not in the original product, If it isn't the original product, then it means it wasn't actually introduced during the processing steps, which can be essential to tracking down exactly where those potential sources of contaminations may be. So in our experience, we've done a lot of use of what is called endothelial progenitor cells. Now, these cells are derived from circulating monocytes, and this is an autologous cell product. In our case, what we're doing is an apheresis procedure. It's a monocyte collection of an apheresis procedure. If you're not familiar with apheresis, what it is that there are two lines put into the patient. The first draws out the blood, and the blood is sent through a device. This device, in this case, is a Terumo Optia, which is an apheresis machine that has a centrifuge in it, and it begins separating the blood by centrifugation and by flow, so that you can get at least a partial density gradient separation, and you can pull at the lymphocyte layer and hopefully more towards the monocyte layer. So you would get a product that is markedly enriched in monocytes. You're still going to get a few red cells, a few platelets, and other lymphocytes. But you want a product from the patient that's markedly enriched in monocytes. The rest of the blood, of course, is circulated back into the patient to the second line. And the anticoagulant used is sodium citrate, which does not do much harm to the patient. That just causes some tingling and whatnot, and is actually the patient can be overcome with calcium, because basically all the sodium citrate is just chelating out the calcium to inhibit the coagulation. So collection of the sample, we then do a biosafe cleanup of the sample, which is basically a density gradient, another density gradient purification step with phycol in this case, but it's done in a closed system. And that allows us to at least to move the granulocytes and most of the red cells. We then plate these cells down and grow them in our isolators for 7 days, and we get what is typically termed these endothelial progenitors, human endothelial progenitor cells, which really are a culture-derived monocyte of monocyte origin. They all start with CD14 positive. But we can then transfect these cells with eNOS, and this becomes a very potent and effective therapy for us. We've used these in both pulmonary hypertension and in the treatment of post-myocardial ischemia, which is the ENACT trial, which we've just finished. And these are the cells take on a very dendritic form in culture after 7 days, with large dendritic processes being shown in this distinct morphology. And in fact, the distinct morphology is one of our QA points before release of the cell product. At the end of 7 days, we have to see this distinct morphology, or we think that something different has gone on in the cells. But one of our problems with this is that the apheresis monocyte yields can vary widely and show poor correlation to EPC yields. So we've done a lot of studies now to look at the monocytes coming out. And surprisingly, when you look at myocardial infarction patients, we can get a lot of monocytes out. That's what some of these earlier ones up here are. We'll look at more of the healthy volunteers. We get a lot lower numbers of monocytes coming out. But we were looking for correlation between the monocyte count and the EPC yield. We get very poor correlation. So there's something else going on. And we've had to dig a lot further. And what we're actually showing is that when we take different cuts across the apheresis in terms of the timing of the apheresis, the EPC yield goes up the further we're into the process. So the longer the run, in other words, the more blood volumes that go through the machine to collect the monocytes, we're starting to collect a different, qualitatively a different type of monocyte that seems to adhere better and actually do this transition into an EPC better. So that's something we've had to work through. So we always now insist on very much longer runs on the apheresis to collect these what we believe are the marginated monocytes, which only come out later in the run. And this gives us a much more consistent product in the end, and a product that we're able to actually grow more reproducibly meet our target dosing and reproducibly get the morphology and the potency that we need in the end cell product. So just as from this point of view, I would argue strongly that spending some time and energy and money on actually looking at the collection of the cells, if it's an autologous process, can go a long way to developing a much more stable and sustainable cell therapy product in the end. We've also done similar tests and look at what's correlating to the yield of MSCs from bone marrow and bone marrow donors. In this case, we're looking at the MESCAMS trial, which was just completed. It's an autologous cell therapy trial taking bone marrow from multiple sclerosis patients and then infusing the mesenchymal stromal cells that arrived over the course of about a month. Really, we grow them up, freeze them down, and then maintain them, and then they're thawed and infused either at the primary time point or 6 months later, and the patients are randomized to receive either at the primary point or 6 months later. But we've started to look at what's correlating to outcome in these cells. And the first thing we started to look at was the nuclear cell concentration that we're getting from the bone marrow draws, which is somewhat variable, but it tends to be a modest correlation with age and a modest correlation, pretty good correlation with the weight of the individual. And there seem to be many more nucleated cells coming out of males than females. However, really the big data that was the most correlative to the output of the MSCs in terms of the number of MSCs we're able to grow was the STRO1 positivity. Unfortunately, STRO1 positivity does not correlate well at all to the nucleated cells in the original product. So really, again, it goes back to this idea, it's not the total maps that you're getting, but there seems to be a quality of products, or it's just the specific phenotype of the cells that you want to collect from those donors to actually get the highest yield, in this case, out of your product. And in this case, that seems to correlate very well with STRO1 positivity in those initial bone marrow samples. So again, that starting material becomes very important, and spending some more time characterizing your starting material, making sure that you can get it in a very clean manner so they're not inducing any infectious agents into your processing, but also making sure that you can get one that is most efficient in developing the cell product at the end, can save a great deal of time and effort and likely produce a much more reproducible product with more likelihood of clinical success. The other thing to touch on is that in many of the allogenic trials, what you might do is start banking the cells, and cell banks have a specific regulatory definition. So when you start talking about a master cell bank, make very sure that the regulatory agencies knows very well what a master cell bank is, and they have well-defined testing protocols for those master cell banks. The master cell bank must be tested with both in vivo and in vitro and human virus testing. Now, there's in vivo and in vitro means that you have to have some animal model for the virus testing. In vitro means it's on cells for some of the virus testing. So they're very long-term tests. They take a long time to be completed. And then but they're not specific to any specific adventitious agents, which is an advantage. Then you can go back and do very specific testing, and these could be quantitative PCR, for example, for specific viruses. And there's a number of specific viruses that the regulatory agencies would like you to test for. And of course, on top of that, you'd have to have full bacterial, fungal, mycoplasma, and endotoxin testing. And you need to have identity of the cells. You want to make sure that you have a pure cell bank, that your master cell bank that you're talking about, that there's nothing in there that looks different, that may cause problems. So that's identity and purity. And then you have to have some activity testing at the master cell bank stage. Then when you want to use that master cell bank, typically what you do is you develop a working cell bank, which is the next stage, a stage immediately prior to developing your therapeutic product. But even in this this working cell bank, you then have to go back and do the in vitro virus testing. And the reason for that is because as you get closer to the total lifespan of the cells in culture, viruses can begin to emerge again. So the viruses can emerge at very end stage when the cell becomes reaching senescence. So you need to really establish this virus free not only at the very early stage of banking the cells down, but at the later phase too. And then, of course, you have to repeat your bacterial, fungal, and endotoxin and do some limited identity testing, again, to confirm that the identity of the cells has remained the same while you're moving through the bank. So developing banks in allogenic is very costly. But the advantage is that you can then go back and draw multiple doses from those banks once they are actually developed in such a manner. Now, moving on to the ancillary materials, and let me just define for you what the ancillary materials are. They are not intended to be present in the final product. They're materials used as processing and purification aids or agents that exert their effect on the therapeutic product. There's a number of them we use in cell culture, of course, all your culture media, your even reagents that you may put in to enhance the growth of the cells or stabilize the cells, anything that you're using to to purify the cells. For example, if you're taking tissue and doing enzymatic extraction of the cells from the tissue, you need to use those enzymes are part of your ancillary materials. And these are distinct from excipients because excipients are intended to be in the final product formulation, and they are often included to enhance the stabilization or the delivery of the therapeutic product. A big one that we use in most of our cell therapy products is human albumin, because we find that the human albumin helps stabilize the cells, stop them from clumping together, and stop them from sticking to the plastics from which they may be delivered in. So you don't want to lose all the cells in the tubing set or in the syringe when you're delivering them to the patient. And the albumin has a big effect there, and also has a big effect on the viability of the cells. For example, when we're delivering them down long catheters, such as a cardiac catheter, we find there's extensive amount of cell loss and cell death if those plastics in those catheters are not coated with albumin prior to the delivery of the cells. So all of the ancillary materials need to have a strong qualification program for them. You need to know the source, the supplier, the grade, and the concentration. You need to qualify the reagent suitability so that it performs as expected, and you have to ensure the reagent quality. And we'll talk a little bit more about that in the next few slides. So the first thing that you should do with any ancillary materials that you're using in your cell therapy product or cell and gene therapy product is do a risk assessment. So there are a number of risk assessment profiles out there. This is taken from the United States Pharmacopoeia, and if you're not aware, the USP in 2019 has just come out with a whole new general chapter on cell therapy, covering cell therapy, cell-based gene therapy, tissue engineering, etc. It's very broad, very encompassing. It's a marked change from what they've had in previous years, and it represents about an update that encompasses the past 5 years. So it's I would urge you to look at the USP if you need to look at something prescriptive that's going to tell you how to do these things and in what manner to do them. So tier 1 is low risk. And we try and push as many of the materials as we can into the low risk category. So if we can find something that we use that there's a reagent grade that we may be using in the lab, if we can find it as a drug product, as a licensed drug product, all of a sudden that becomes a low risk product. So they're because it's a highly qualified material, it's licensed, the regulatory agencies and you know exactly how it may behave in humans, if there are any residuals involved in your product, or it may be an approved drug or medical device. So these are no-brainers. If you can get all of your products pushed into low risk, it makes a big advantage that you have to do much less. Tier 2 are low risk as well, and these are materials that are well characterized, suited for drug or biological or medical device manufacturing. In fact, the design of the manufacturing would be taking this in mind. In other words, it's not a reagent grade product. It's not manufactured only for diagnostic use, but it's manufactured specifically for the use in the manufacture of therapeutic biologics, cells, or proteins. Now, this, of course, excludes most animal-derived products, and unless they are specifically designed and tested to do this. And some are. Some may fall into the licensed biologic that you can then use. The moderate risks require a higher level of qualification, and they're the ones that we've 10 years ago, almost everything we were doing fell into this moderate risk because we had very few suppliers that were specifically manufacturing the products that could be used in clinical trials. Everything was labeled as for research use only. So these require a higher level of qualification. And they're often, as I said, they're produced for in vitro or diagnostic use or for laboratory use, not for use in further manufacturing into humans. And then there's high-risk products, which require extensive qualification prior to use in manufacturing. And I'll get into these on the next few slides because the USP does give some very nice tabulated examples of what these may be, and then also what you're supposed to do if you're doing full GMP. Now, I think what we need to define here, as I stated early, we may not be doing full GMP when we go into an early phase cell therapy trial, but we have to keep these things in mind that this is what really is necessary as we would be moving to that final licensed product. But we need to understand that if we're not doing everything, we have to have a rationale why we think it would be safe otherwise. So again, we don't have to spend a lot of time on these low-risk, highly qualified materials intended as therapeutic, drug, or biologic. If that's the case, you probably use a drug master file. More likely, there's even more data than that, and you might not even need to cross-reference the drug master file if it's a well-used a product such as an injectable antibiotic that you're using in your system. You may not need, it may be fine just to know that it's licensed and you're regulatory, particularly in early phase clinical trials. You may just have to show that the licensure and to Health Canada and the certificate of analysis and the lot. Now, however, when we start to use these products, we have to assess lot-to-lot performance. You can imagine something like albumin or other products that we're using, even though it's a licensed product, there may be variability lot-to-lot, and that lot-to-lot performance may have a big impact on your therapeutic product in the end. So you really need to handle that lot-to-lot, and you need also assess whether or not you're removing it from the final product and how much you're removing from the final product, and then assess. Often in almost all regulatory submissions I've seen, as we come to later phases, this is what Health Canada would be asking you for, is what are the residuals of those ancillary agents in your final product? And if there are residuals there at such a level, what impact could that possibly have to the patient receiving them? So you also need to look again as a stability assessment. Now, this is different from the stability that the drug manufacturer may be doing, because you may be actually diluting this down. You may be using it at different concentrations and storing it at different concentrations. So you'll have to do your own stability assessment to make sure that you are actually storing the product in the facility in a manner that it is safe. It's not going to become a problem in terms of sterility of the product, and that it's also going to be as potent as what you want it to be, or as stable and as reproducibly potent in the systems that you're using it in. So examples that the USP gives, cell culture medium additives such as insulin, some organ preservation fluid, which would largely be buffered, which are specifically used for those things, and licensed for such use. We use a lot of human serum albumin, as I talked about. It can be a cell culture medium, but we've also used it as excipients in our final cell products. There's sterile fluids for injection, implantable biomaterials that would be licensed already, RNA or used for inhalation or injection, antibiotics, as I said, except the beta-lactam antibiotics, of course, you'd want to stay away from because they have a very high probability of inducing immune reactions in a subset of patients. So if you can avoid those, it's much more acceptable. Some of the injectable monoclonal antibodies, of course, you could use, and injectable cytokines are already approved for use, would be ideal to put in your product as opposed to the research grade ones. Vitamins, nutrients, IV bags, and the tubing sets, etc. Those all fall into tier 1, which is relatively low risk, and the more things we get into tier 1, the easier it is for our processing. Unfortunately, most of the stuff that we have probably falls into nowadays is starting to fall into tier 2. These are well-characterized materials with intended use as ancillary materials, and they are produced in compliance with GMPs. No word on GMPs here, because although your manufacturer says it's GMP, that often is a meaningless term unless it's a licensed product. If it's a licensed product, it's manufactured according to licensure, which would include a GMP. But if it's a non-licensed product, the term GMP becomes a little bit flaky, and often just means that they have a different system. They may have a different line that they're manufacturing on, but sometimes that may or may not be the case. You'd have to confirm that. There's no regulatory obligation for them because they have no regulatory submissions stating that it is GMP. So the GMP manufacturing you have to take with a grain of salt, and you'd have to confirm that the manufacturer is in compliance with as much of the GMP requirements that you need them to be in compliance with. Whether or not that's separate lines for manufacturing and complete segregation from other things that may be high risk in the manufacturing plant that they're doing these things in. That's up to you. I think that's up to you to decide on the risk based of the product. So it's a little bit back to the sponsor here as to what would be a GMP in terms of some of these things. But really pushing them towards that more GMP compliance is really an advantage. And many manufacturers are now at least labeling things as GMP and have then gone back and done some things to help you assert that they have developed a higher quality standard than the regular reagent grade products that they produce. In this period, a lot of recombinant growth factors are cytokines. Now, they may be coming from animal sources, but they also may be, but typically recombinant ones would be coming from bacterial sources. Where they're manufactured, how they're purified, really needs to be is something that's very difficult to discern. So you have to go back, and often the issue is recombinant growth factors are going to be part of some of the media kits that you're getting from one manufacturer who's sourcing it from a source that you're not aware of. So you really have to dig very deep and go back to the original sources to confirm that they've done their due diligence to make sure that these products are safe to use in further manufacturing. And then you do need to know the levels, roughly the quantities of these growth factors in the media, because these are things that the regulatory agency is going to be asking you is, how do you know what the residuals are going to be in there? And if you don't know what the original is, it's very difficult to determine what residuals may be in your final product. So they can be involved in immunomagnetic beads, human AV serum, cell culture medium additive, but again, many of this we get licensed as a licensed product. Some of these may be USP grade products that you're using, so you have to make sure that they're of high enough quality. We have sterile process buffers. Again, often those are USP grade. Biocompatible polymers, again, proteolytic enzymes. We'd like to see that they're all manufactured for what they're used for in terms of all of these reagents. So again, you can have a drug master file that you can really cross-reference. So in these cases, it becomes essential to have the drug master file. You may not know what's in that drug master file, but at least the regulatory agency will get a look at it. It's best, of course, to enter into a non-disclosure agreement with the manufacturer if you can, so they can then supply you with what's in their product, so you can then make your own risk assessment. Because you don't want to get caught just finding out that it's a very high-risk product when you put your CTA in, and Health Canada has reviewed the drug master file and has decided that there's a significant risk here that you're not mitigating. All of these should come with a certificate of analysis. You have to make sure that you're checking off the fact that the certificate that you've checked that the product meets the certificate analysis. So typically you would look at one of the items on the certificate analysis, such as sterility, for example, and make sure that it does meet that classification. You have to look at lot-to-lot effect, because again, these aren't as well characterized as a licensed drug. So you need to look at performance lot-to-lot. And this is something, as you're moving along, particularly in autologous cell therapy, you have to be very careful. It's because lot-to-lot means patient-to-patient variability. You have to assess whether or not you're removing as much as you can, as best you can. You have to assess that we remove these ancillary agents from the product. Again, the stability that we talked about before, and this is why I just talked about the testing certificate analysis. But also what's required here if you're doing full GMP is a vendor audit. Now, for many of these tier 2 products, we wouldn't necessarily do an on-site audit, but that's likely required if you're doing a licensed product. So in the early phase trials, we may do more of a vendor initiated audit on their own. So we send them a bunch of questionnaires, we talk them over the phone. If we are comfortable that the products are manufactured at a highly reputable company under good conditions. If there's any question at all, then I think a site visit is in order to make sure things are being manufactured appropriately. The vendor audit becomes essential once you get from tier 2, 3, and 4 items. You have to make sure that the vendors are qualified to manufacture what you're doing, and that's a sponsor requirement. So if you're not doing GMP unless you're sure that those vendors are actually providing you with the quality that they ascertain. So the tier 3 products are moderate risk materials, and they're not intended for use as ancillary materials. They're frequently produced in vitro for diagnostic use or reagent grade materials that you might use in the lab. So in those could be, again, your recombinant growth factors or cytokines that are lab grade. You could have tissue culture media that may or may not be produced to full cGMP. If you can get it manufactured GMP, it would be better. And then specifically, some of the monoclonal antibodies or process buffers, whenever you can get them manufactured at a higher level, it would be much better. And again, novel polymer scaffolds or hydrogels or enzymes or other things you might be using, such as proteolytic enzymes or some chemicals that you might want to add that might be purified. So in this case, you have to do a lot more quality assessment of the products and perhaps risk mitigation efforts. Just as what we were talking about before, we're down to, again, the vendor audit becomes much more critical here. There have been a number of incidences where some of these products that have come in and the regulatory agencies now for them, some of the heparin issue that had just come up in the past 10 years has raised significant red flags with the regulatory agencies, so they will be aware of it. So understanding and understanding where the product is coming from and having some form of audit system in place to look at the vendors meeting those quality assurances that they may give to you is essential. You would be encouraging them to upgrade manufacturing process for material to a GMP-like process. And again, we talked about that. So if we can push them to that, and many of these reagents are now coming that the vendors are claiming are GMP. So that's good. It's at least moving to higher quality standards, but you have to make sure that they're doing that. And you want to develop stringent internal specifications for these products as they're coming in. You may want to do some of your own advantageous agent testing on them. For example, if you get a lot, we've done that before with lots of serum or lots of other biological products, we would send samples of that lot out for advantageous agent testing and our own bacterial testing just to confirm that they're sterile and meet at least specific advantageous agents that are not in the products as you're using it. And again, lot-to-lot biocompatibility, cytotoxicity, or adverse agent testing are needed in many cases, and that's what we're talking about there. So a much higher level of qualifications when you're starting to use the Tier 3 materials. And again, this level of qualification may go up the further you go down, the later phase you are in your clinical trial program. So, Tier 4 are very high-risk materials. FBS is listed as a Tier 4 material, and that's largely because of the risk of bovine viral disease that may be in FBS, as well as fungiform encephalitis. So you have to confirm that you're getting it from a clean source, and then you're going to have to do a lot of testing on it to make sure that you can use it. That's not to say that you can't use FBS, and FBS has been used in a number of clinical trials. It just means you have to do a lot of quality assurance to make sure the product is clean and acceptable for use in your manufacturing system. So you have to particularly want to verify the traceability to country of origin. That's particularly true for things from bovine sources, so that you're not getting it from a region that's endemic with TSE in the herds that it may be coming from. And so origin is qualified as safe respect to TSE. And usually what you would like as well is some sort of reporting system back to you so that the vendor knew if something did spring up in their sources, they'd immediately notify you. You can pull the product that came from there, and you can start, again, looking for a cleaner source. And again, you're probably going to be doing some advantageous agent testing on these materials to make sure that they are clear of relevant animal sources of viruses. That is, if you can't do some viral mitigation strategies that you may also need to apply to some of these materials. So a lot of these are animal-derived extracts, polymers, scaffolds, FBS. We've talked about purified enzymes, ascites-derived antibodies or proteins, which are not well characterized, maybe made in a mouse or something. Animal human cells used as feeder layers. You have to be very careful with these cells and make sure that the cell culture substrate. And so again, this is where you go back. It might be from a master cell bank, and you have a well-qualified master cell bank if you're using it as a feeder layer. So it almost has to be as well qualified as your final product. And chemical entities with known toxicities that you might have, and you've got to make sure that they then are completely removed and you're using them properly. So managing the risks of ancillary materials. So as I said, again, I'd like to just stress full cGMP requires the complete adherence to what I just showed you, which is the USB guidance. In early phase product trials, this may not be the case, or you may look at those things, understand what needs to be done, and do as much as you can that is practically possible, and then submit that to the regulatory agency and see if that will be acceptable to them. Because you're balancing the risk here, and you need to balance the risk-benefit profile in their whole clinical trial. So often doing as much as you can for the money that you have can work, as long as you're very careful and you think about it from an evidence-based approach, and you think where the high risk is, and you do your best to mitigate those risks as much as you can. So this often involves working closely with suppliers, having site visits or audits if you can, and having quality agreements in place. And those quality agreements, okay, not only are the supply to you, but also if there is a problem, because again, these aren't licensed products, there's going to be no recall of them. But if there is a problem in processing that they have come up with in the plant, you want to be notified by them and then come to some terms as to whether or not you withdraw that from your manufacturing or how you move ahead to make sure you have clean products as you move on. You want to, if possible, review all the source documents, C of A's for all clinical source reagents. Again, you can do this in a vendor audit as well. You review all their SOPs during the development and the infrastructure that they have in place to manufacture the product. If you can't do that, you may have a vendor self-audit where you ask a series of questions, and those questions are answered by the vendor, and you're relying on them, and that may be acceptable for the earlier phase trials. You want to do regular in-house testing of the materials, but you use them for sterility and stability, and for the ability to do what you want them to do in your product. Establish your storage protocols to establish working shelf life. Again, working shelf life in the manner in which you store it and use it in developing your product. Define the potential risk source to lowest risk, country, batch size if you can, or the lowest product. If you can find, again, a licensed product, that's much better. Consider the use of alternative processing methods. You might have to either delete that ancillary material from the processing. If there's another one that might work better, that might be a licensed product, you should consider that up front. You might also want to consider things that might mitigate those risks, sterile filtration, use of antibiotics in some parts of your process if high-risk materials are completely unavoidable. And then, of course, you always are going to have to measure and find some form of assessment as to what residual level is in the final product, so you can understand fully what's going into the patient when you formulate that final cell therapy product. So just as a quick primer going back, the WHO does have tables on tissue infectivity distribution in TSE. TSE is a big issue in the past 20 years or a little bit more than 20 or 25 years. So it has caused some significant issues in some, for example, human dura matter transplants. There was a significant outbreak of TSE and Trussell-Jacobs disease in a number of patients who had those implants, so there was some legitimacy involved. So when we look at the high infectivity tissues, it can tell us, things that we might want to be very vigilant about. Things coming from the brain or spinal cord of a cow, for example, would be very high risk, and you'd want to make sure either not to use them or to find alternatives, or to have a very good purification process where you absolutely can confirm that you're coming down to only the product that you want. And I'll show you an example of that. There are other tissues that are low infectivity, and if you go on in the WHO classifications, there are even tissues from bovine that are almost no infectivity risk. So that's the first area that you might want to look in terms of classification, is what is the risk of the starting material before you even go in. And as an example, let's look at aprotenin. Aprotenin as a serine protease inhibitor was used constantly in the lab, and we might want to use that in many of our activity assays or in our cell isolation assays, because it inhibits plasminogen, it inhibits trypsin. So if we're dissociating tissue, we may want to stop that immediately with an aprotenin. However, if we get it from the lab base, it's going to be a tier 4 risk as a laboratory reagent. However, luckily, it was developed by Bayer as an injectable drug, crasolol. Now, it had some problems in that its clinical effectiveness was questioned, and whether or not it had a higher mortality rate than other plasminogen inhibitors. So it was withdrawn from the market, but then put back on the market in 2012. And that's fortunate, because here's a purification strategy where they spiked in either bovine-specific viruses, parvo or viral diarrhea virus, or prions, and showed that various phases of the extraction process, which are represented by plus here, do very good at removing those products from the final aprotenin. So this is how it got passed and licensed as a drug, but you have to appreciate that the licensed drug has gone all throughout this validated testing and validated processing. The reagent grade may not have gone through those types of processing. So you have to distinguish getting the licensed drug, which would be very likely to be very safe, from other reagent grade aprotenin that you might be using in your process. So just quickly, I'll just go over some of the types of materials that we used in some of our processing. Culture medium for the MSCs, we've used Nutristem cell culture medium, which would be a tier 2, tier 3, because it does have pharmaceutical grade. It has human products, It doesn't have animal products in it, but it does have some human products in it, and it does have growth factors in it that we don't know exactly the source of those growth factors. So they're like either tier 2 or tier 3. We have to dig into that. We've had several conversations with the company. We have a drug master file with Health Canada. We've got a confidentiality agreement in place with the biological industries. So we do know what's in it, and we've tried to mitigate as much of those for the early phase trials as we can. If we move on to later phase trials, we have to probably do more of a vendor audit and find exactly what's going on there. We look at things like platelet lysate, which we can then use a DMEM and platelet lysate max or platelet lysate gold from Mill Creek. It's likely a tier 2. It's a human product, but it has been very well characterized and is manufactured for use in manufacturing. And in fact, it's got you can even buy it with a viral reduction process in it, so that has been applied to it. So it's a clinical, you can get it as a clinical grade product. Fibronectin from Roche is tier 3. It really isn't manufactured for use in manufacturing. It's manufactured for research grade, but they do push it to some form of GMP grade, which is good, which means it's reproducibility on the product. They do an extensive amount of adventitious agent testing. So it's, I guess, between tier 2 and tier 3 because they do a lot of the testing for you, and then you probably have to do some of the testing in-house. Triple LE Select, it's a tier 2 because it is manufactured for use in further manufacturing. But again, you have to do some qualification of that, make sure it's okay batch to batch. And then some of the other ones are tier 1, tier 2, the cryostorage mediums. Just looking at the media and why it's important, when we grow out human MSCs, and this is again part of the MESCAMS trial or other from healthy donors manufacturing these cells, the different medias have a huge impact on cell growth. We can go from, in 6-day period, from 7 population doublings, from 1 population doubling with a classic FBS DMEM, up to over 7 population doublings, which has a major impact on the quality of those cells and may really major impact the safety of those cells, because we know things like tissue factor can be expressed based on the high degree of proliferation of the cells. Cancer cells, which are highly proliferative, produce a lot of tissue factor protein, and tissue factor has been shown to be growth factor stimulated. So these types of things can be very important in terms of the quality of the product that you're producing. I go back to the EPC manufacturing. We're using UBM2, which, and the single quats, again, this could be tier 2, tier 3, because we don't know tier 3, because for the single quats, we don't know exactly where those growth factors are coming from. I've talked about the human fibronectin from Roche, where they're now manufacturing GMP grade. And then the pooled human serum, again, tier 2, tier 3, this is, again, well tested and GMP manufactured, so we do get a high quality grade of the human serum there. The mini circle we have manufactured ourselves specifically for use in the trials at tier 2, and I can go through some of the extensive characterization we do there. Again, similarly with our polyethanolamine, which is our transfection reagent, is a tier 2. It's specifically manufactured in GMP source manufacturing for use in manufacturing of clinical products. And then we talked about before some of the other products that are listed here. So some of the issues we've had, for example, we're dealing with Lonza for a long period of time with the media. They had an inability to supply us. They had lot-to-lot variability, significant impact on cell phenotype. Some errors in manufacture, and we had sterility issues. And these are the notes that we came back. Lonza came back to saying the Walkersville site had a significant sterility issue problem. They weren't recalling it because it wasn't a licensed product, but we were just notifying you that that was the case, and you have to make your own decision as to whether or not you'd want to use the product moving ahead. And they stopped manufacturing. And they said, well, we can manufacture not in our Walkersville site, but we can manufacture in Belgium for you. But then we get a letter from the Belgium site saying the same thing. So it was very sometimes, and then we did an audit of Lonza in Walkersville, and we had some concerns, but they allayed most of our concerns during the audit. They had made some significant changes, but it took them at least 2 years before they were able to manufacture again, which puts a big hole. In the meantime, we had to find other sources of manufacture. And this just goes to show some of the basal media issues that we have. The basal media combined with the type of serum we're using, we can get very different results and different expression of, for example, CD14 on our cell product. So the media goes a long way to defining what the product is, so it has to be well qualified to begin with. Similar with some of the fibronectin reagents we've had. This is an attachment solution, which is 100% fibronectin. However, we can see that there's a number of bands when you run it just on a simple protein gel, whereas the fibronectin from Roche is really just principally a single band, and that's also reflected in the results that we're getting, where we get much better attachment with the roach fibronectin. So again, just quickly going over how we manufacture from a GMP and GMP source manufacturing for our DNA. We've contracted Aldevron to do this. We have a full manual of what they're doing that we can submit to the regulatory authority. And then we have the full certificate analysis, which is several pages, showing exactly what we're getting in terms of how well qualified the DNA is. This can be very chemically defined, but the degree of supercoil that we have in the product can really have a big impact on our transfection rate. The higher the supercoiled product, the better transfection we're getting. So again, the quality of those products can have a big impact, and you want to make sure those products are high quality and that they are reproducible lot to lot when you're manufacturing them, so they are giving you what you want in the final cell product. When we go to vendor audits, what we try to do is a vendor classification system. We have a whole audit SOP process. We look at the quality level assigned that undergone prior to vendor assessment purposes of SOPs. We have on-site audits. We have an on-site audit checklist. We go through those audits. We give them a quality rating, and then we qualify the vendor. This is the Lonza facility after it's been upgraded, but this was interesting. This was taken from the contractor's website, showing what a good job they did, but basically saying what a mess it was prior to them coming in and fixing it, which really tells you, and that was at 2017. So prior to this, it can tell you that's where they were manufacturing in plants that really needed significant upgrades. On the flip side, I've been to facilities where we're now having most of our media manufactured with the Bioconcept, who have in Switzerland, which has an extremely modern plant. As soon as you walk into these facilities, you really understand what the highest level is. This is all automated processing, robotic processing inside an isolator, where for bottle filling and then manufacturing is all done in the cleanest clean rooms I've ever seen. So, and this is not often that you see this in media manufacturers, because they are at a very low manufacturing cost to keep the price down. But this Bioconcept plant actually manufactures, supply the drug industry as opposed to just a regular R&D services. So it's at a very high level, and it's very impressive. So just to summarize, there are numerous challenges in the manufacture of clinical cell therapy products in the academic setting. These are often manageable. New technologies such as isolators and closed systems may provide significant advantages in academic settings. Ancillary reagent suppliers are still catching up to provide GMP quality improvements that are evident, but diligent planning and oversight is required to reduce the risk involved in the use of many of these ancillary materials. Maintaining the focus on evidence-based protocols proven to enhance the safety and quality of the final product should be a primary goal. And I think I'll end there and just thank, there's a bunch of people involved in the GMP facility here who helped out, and the clinical trials were done in collaboration with a number of different investigators. Okay, I think I'll stop there, and I think I can entertain questions.
Just an admin note, if anyone has a question, you can either unmute yourself in the chat, or you can type in a question in our chat box. I'm posting a link to a survey from SurveyMonkey, so you can let us know what you thought of the webinar. And that should also pop up so you can see where to ask your questions if you have any. I have a quick question to get started with. It seems like it could be a bit daunting to get started on the audit process if it's something that you've never done before. So how did you go about developing sort of some of the SOPs that you used when you went to the facilities to check and see how they were doing, and how would you recommend someone else get started on that?
Well, we try to draw from whatever resources we could find. Obviously, there are some good resources out there. As I stated before, the United States Pharmacopoeia does help with giving some ideas of what should be present and what you should be looking for. Some resources such as the PAC facilities have some SOPs online. I can't remember if we drew from any of those, but also FACT, I think, has some documentation along what the expectation for vendor audit should be as well. So we draw from as many resources as we can. And then part of it, unfortunately, is just experience, because before you go and visit some of these sites, you don't really know what you're going to be expecting to see. And some of them can be pretty bad. And then you have to sort of define in your own mind whether or not showing you what they think are state-of-the-art facilities, which actually are Mettler balances there, where the front is taped together, is actually an
Oh no.
acceptable, process or not. Again, the range in what's going on here is wide, and also you need to put everything in context, and that maybe that's not that bad if everything is what you're seeing in terms of how they're manufacturing might not be that bad if everything is endpoint sterilized at the end as well.
Right. So I have a question from Craig. How do quality agreements cover your due diligence versus a full audit of the vendor or manufacturer?
Well, I don't think a quality agreement would negate your due diligence as doing a vendor audit. It would help out. I think you have a due diligence obligation. What I think what comes into due diligence in this case is what the expectations and the industrial standards would be. And since these are well documented now that vendor audits are essential, parts for full GMP manufacturing, I think you have an obligation to at least perform some form of audit. And the quality agreement may help in the earlier phase clinical trials, but I think the quality agreement I think is essential more in that there is a notification that they are continuing to supply the quality that you started with. I think that's really essential in the quality agreement, that it's not changing as you go through your long-term clinical trial, or that at least you're notified if it does change.
Right. So if anyone has any other questions, I think we might wrap up just because we're going a bit over time.
Okay.
But thank you very much for taking the time to do this. I thought it was very interesting, and hopefully we have a comments also. Thanks in the chat box. So yes, we will see everyone at our next webinar, and thanks again.
Okay.
Bye.
Thanks.